The topic that I'm going to discuss in this video is about PCR. PCR stands for polymerase chain reaction. PCR is a technique used in molecular biology to amplify a single copy or a few copies of a segment of DNA across several orders of magnitude, generating thousands to millions copies of particular DNA sequence. PCR was developed by Kary Mullis in 1983, and this is a very common technique used in clinical and research laboratories for a broad variety of applications. The basic principle of PCR technique is based on the enzymatic replication of DNA. In PCR, a short segment of DNA is amplified using primer-mediated enzymes. DNA polymerase synthesizes new strands of DNA complementary to the template DNA. The DNA polymerase can add a nucleotide to the pre-existing 3'OH group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' end of the DNA polymerase. PCR possesses high specificity and enables exponential amplification of the target DNA. At the start of PCR reaction, DNA is double-stranded. This double-stranded DNA acts as the template. The next component that is required for PCR is DNTPs, that is deoxy adenine triphosphate, deoxy guanine triphosphate, deoxycytosine triphosphate, and deoxythymine triphosphate. Next component involved is DNA polymerase. For this, Taq polymerase is used. It is thermostable and it does not denature at very high temperatures. And finally, oligonucleotide primers are required. These are the short stretches of single-stranded DNA complementary to the 3' ends of sense and antisense strands. Three major steps are involved in PCR. The first step is denaturation. Denaturation occurs when the reaction mixture is heated to 94 degrees Celsius for about 0.5 to 2 minutes. This breaks the hydrogen bonds between the two strands of DNA and converts it into a single-stranded DNA. The single strands now act as a template for the production of new strands of DNA. This temperature should be provided for a longer time to ensure the separation of two strands. Next step is annealing, in which the temperature is lowered to 54 to 60 degrees Celsius, during which the primers bind to their complementary sequences on the template DNA. The third step is elongation, in which the temperature is raised to 72 to 80 degrees Celsius. The bases are added to the 3' end of the primer by the Taq polymerase enzyme. This elongates the DNA in 5' to 3' direction. The DNA polymerase adds about 1000 base pair per minute under optimum conditions. These three steps are repeated 20 to 40 times in order to obtain a number of sequences of DNA of interest in a very short period of time. The number of DNA fragments amplified exponentially, and the number of copies keeps on increasing by the number of cycles. The result of PCR can be observed in aqueous gel electrophoresis and visualizing it in UV trans illuminator. Thank you.